Microsporogenesis is nothing but the production or development of male gametes, that is microspore or pollens inside microsporophyll. The male part of a plant, that is stamen, typically have long slender portion called filament and an upper sac-like fertile portion called anther. The stamen arises from the thalamus, and inside the anther there is a mass of meristematic cells. As you can see in the image, it is the transverse section of mature anther. You can see four lobes, that is Microsporangium, one on each side. At the beginning, there will be four archisporial initial cells which divide to form two types of cells, that is primary parietal and sporogenous cells. The primary parietal cell further divides to form four types of cells, that is epidermis, endothecium, middle layer, and tapetum. The epidermis is the outermost layer formed of barrel-shaped parenchymatous cells with no intercellular spaces. They are protective in function. The next layer is the endothecium that lies inner to the epidermis and is also made of single-layered parenchymatous cells. It is also protective in nature. They have callose thickenings or fibrous bands inside them which makes anther elastic and rigid. The middle layer is made of 3 to 4 layered parenchymatous cells and it is filling tissue in anther. The innermost layer is tapetum, which is present next to Microsporangium and nourishes the developing pollen, so it is also called as nutritive tissue. It also helps during the dehiscence of pollen grains. The tapetum produces callase enzyme, which degrades the callose present in endothecium. It also produces yellowish bodies that coats the developing pollens with sporopollenin, which is the most resistive substance to the physical atmosphere. Inside Microsporangium are the pollen grains. At the time of dehiscence, the anther breaks at the stomium, releasing pollens. As I have told you before, at the beginning there is archisporial initial cell that divides to form primary parietal and sporogenous cell. We have learned about parietal cell. Now let us study the sporogenous cells. The sporogenous cell further divides into pollen mother cell which develops into microspore mother cell or MMC. The MMC undergo two meiosis. The first meiosis produces a dyad and the second meiosis produces a tetrad. Based on tetrad cell arrangement, it is further classified into 5 types: tetrahedral, isobilateral, decussate, T-shaped, and linear. This haploid tetrad is nothing but immature pollen grains. Each tetrad separate out and form 4 pollen grains. Each haploid cell gives rise to a single pollen grain. So that's all about the pollen grain development or microsporogenesis. So let me summarize this. The pollen grains are developed inside the anther. At the beginning, there will be archisporial initial cell that divides to form primary parietal and sporogenous cell. The primary parietal cell produces the 4 outer layers, that is epidermis, endothecium, middle layer, and tapetum, whereas the sporogenous cell give rise to the pollen grains. So thank you for watching this video. Make sure to subscribe to my channel for more contents like this.